Good morning everybody. Myself Sandhya. Today we have to discuss about the topic of bacterial growth in batch culture. Microorganisms growing in a culture utilize nutrients for their growth. As their population is increased, more nutrients are utilized, which result in the depletion of nutrients in the media. Under these conditions, cells control their growth and eventually die when the nutrients get exhausted. Bacterial growth regulated by nutritional environment. When suitable environment is there, that time bacterium is incubated. Its growth leads to increase in number of cells which allow definite course. The growth curve has got 4 phases: lag phase, log phase or exponential phase, stationary phase and decline phase. Lag phase, bacteria adapt themselves to growth conditions. It takes place 1 to 4 hours. It is a period where the individual bacteria are maturing and not yet able to divide. During the lag phase, the bacterial growth cycle, synthesis of RNA, enzymes and other molecule occurs. Length of these phase depend on the bacterial sepsis, culture medium and environmental factors. Then next one is log phase. It takes place 8 hours, sometimes called log phase or logarithmic phase. It is a period characterized by cell doubling. The number of bacteria appearing per unit time is proportional to present population. If growth is not limited, doubling will continue at a constant rate. So both the number of cells and the rate of population increase doubles with each consecutive time period. For this type of exponential growth, plotting the natural logarithm of cells number against times produces a straight line. The slope of this line is specific growth rate of organisms which affect the frequency of cell division events and probability of both daughter and cells surviving. Under controlled conditions, cyanobacteria can double their population 4 times a day. Exponential growth cannot continue indefinitely, however, because a medium is soon depleted of nutrients and enriched with the waste. Then third one is stationary phase. Stationary phase is due to growth limiting factor. This is mostly depletion of a nutrient and or formation of inhibitory products such as organic acids. But unfortunately, widespread explanation of this stationary phase result from situation in which growth rate and death rate have same values. Such an explanation would not be accordance with the observed substrate depletion and also could ever explain rather smooth horizontal linear part of the curve during stationary phase. Death cells as function of time rather unpredictable and very difficult to explain. Another two really logical explanation of the stationary phase is that there isn't anymore enough space for the cells. However, under the microscope you will see that there is still plenty of water between the cells only in an agar colony, which densely packed space is obviously limiting. Decline phase. Bacteria run out nutrients and die, although number of cells remain constant. The decline phase is brought by exhaustion of nutrients, accumulation of toxic products and autolytic enzymes. Sometimes small number of survivors may persist the mouth even after death of majority of cells. These few surviving cells probably grow at expenses of nutrient released. Thank you.